Hello, and thank you for joining us today for this webinar, Exploring Protein Analysis, Techniques and Applications in Bioanalysis, brought to you by Script and sponsored by Aliri Bioanalysis. My name is Janelle Hart, Managing Editor at Sightline, and I will be your moderator today. Before we jump into the presentation, let's go over a few housekeeping items. First, if at any time you are having technical difficulties, click on the Test Your Connection button, and this will take you to a connection checker and frequently asked questions page. If you're experiencing any technical issues, there is a Chat Now function to contact the support team for assistance. Want to enlarge the presentation? Just click the Maximize icon or drag the lower right-hand corner of the presentation window to enlarge. There will be a Q&A at the end of this presentation, so if you have any questions, we want to hear from you. To ask a presenter a question, simply type it into the window on the bottom of your screen and then hit Submit. We'll be answering as many questions as possible during the Q&A session, and if there are extras, we'll try to answer you after the webinar via email. Because we know you'll want to listen to this webinar again, today's session is being recorded and will be available to view on demand in about 48 hours. I will now like to introduce our speakers for today's session. We have Shane Karnik, Senior Laboratory Director of Bioanalytical at Aliri. Shane joined Aliri in 2003 and currently leads a team of bioanalytical scientists conducting projects that support pharmaceutical drug development for both non-GLP and GLP preclinical and clinical studies. In addition, he supports the technical and regulatory aspects of regulated studies. We also have Edward Brewer, Senior Director of R&D, Mass Spectrometry at Aliri. Edward joined Aliri in August 2016 and currently leads the non-GLP programs. Ed has been involved in bioanalytical mass spectrometry since receiving his master's degree in 1986, and his expertise comes from experience within forensic toxicology, large pharmaceutical companies, and contract research organizations. And finally, we have Matthew Hartle, Analytical Chemist at Aliri. Matthew's research interests focus on the application of protein isolation and digestion to the quantification of various proteins by mass spectrometry and liquid chromatography mass spectrometry. Dr. Hartle received his PhD in chemistry from the University of Oregon in 2016. And with that, I'd like to hang things over. Shane, Ed, and Matt, take it away.
Good morning, and thank you, Janelle, for that introduction. I'd like to start this webinar about the analysis of proteins with the basics, and that is, what is a protein? A protein is essentially a linear polymer built of monomer units called amino acids. As it's being synthesized on the ribosomes, it spontaneously folds into a three-dimensional structure, which is considered the tertiary structure of a protein, while that linear sequence of monomer units is called the primary structure. The primary structure amino acids contain a wide range of functional groups, and most of these are chemically reactive, which can interfere with your analysis. Once a protein is formed, they can interact with one another. They can form complex assemblies with other macromolecules. They can self-associate with themselves to form quaternary structures of trimers and quatrimers or dimers. The other thing to know is that proteins come in a wide range of rigidity. Some are incredibly rigid and don't change conformation very much, where others are considerably flexible. And that will have impact on how we go about analyzing the protein and how we break it up through a digestion. Now, proteins are only one component in a complex biological system, and I would like to highlight some of the other classes that the FDA has defined besides just proteins. The first one that I'd like to highlight are antibodies. These are also called immunoglobulins or Ig proteins. They are synthesized in a biological environment due to a foreign substance or antigen entering that environment. The antibody is composed of two main regions, the heavy region, which is constant, and the light or variable region. And that variable region can change to recognize very specifically a small group or cluster of molecules on an antigen. And that group or cluster of molecules is called the epitope. When an antibody is synthesized in a biological system or in some kind of a species, it is highly specific to the antigen that it is synthesized for, which makes it a great tool if we can isolate just that one antibody. The problem, though, is that most species have a large number of polyclonal antibodies as part of the immune response because they're looking at sort of blasting an entire wide array of things. We have been able to get around this by looking at monoclonal antibodies. We isolate a single antibody-producing cell, and then we can produce only that antibody so that all the antibodies are identical to each other. These are often modified so that they become humanized, and that creates a series of antibodies where the heavy region matches human IgG, and that is helpful in a therapeutic sense because it becomes more accepting to the body. Monoclonal antibodies are also useful in a multitude of assays for ligand binding assays, ELISA being the most commonly used one, as well as hybrid LC-MS-MS assays, which we will get into. The next class I'd like to highlight is mRNA. But before I can really talk about mRNA, I need to talk about DNA. So DNA contains the genetic code or the instructions for all the proteins that are synthesized in a living system. However, rather than having a cell take that DNA out of the nucleus and then be translated into the protein or read and synthesized into a protein, it first goes through a process of transcription to help keep the DNA protected in the nucleus of the cell. It is transcribed onto messenger RNA. That messenger RNA can then leave the nucleus and be translated into the protein that the gene or the small section of the DNA is designed for. DNA is not the direct template. RNA is. It also represents only about 5% of all the RNA in E. coli and is considered a heterogeneous class of RNA molecules, unlike other ones that you might see talked about, like small interfering or looped RNA. I really want to mention this particular class because it recently won the Nobel Prize in Chemistry this year for the modification of mRNA for applications in therapeutics, ranging from vaccines to rare diseases. The final area I kind of want to talk about is what the FDA created sort of as a catch-all called biologics. And this is going to be sort of everything else. It's non-antibiotic proteins such as transthyretin, transferrin, or dystrophin. It can range from simple sugars all the way up to some living entities such as cells and tissues. These are on the cutting edge of biomedical research and often are an effective means for very rare diseases that have no other treatments available. So a lot of studies that produce biologics as therapeutics are N of 1 or very small studies. Here at Aliri, we can develop an assay for nearly all biologics, whether it's a protein, a DNA, RNA, simple sugar molecules, but we have a wide range of analysis available to us. When we're talking about proteins specifically, there are 3 main types of assays available to analyze and quantitate those proteins. The first is a ligand binding assay, or LBA. The advantage of an LBA assay is that it is incredibly sensitive, and you can run through it fairly quickly. They can get down to picogram per mil sensitivity. However, the specificity of an LBA assay is limited to the antibody that you have chosen for that assay. And so if you choose something that, for example, grabs immunoglobulin proteins, you may grab more than just your protein of interest, especially if your protein of interest is a humanized version of that protein. To get around this, we can do direct digest LC-MS. This has significantly greater specificity due to the retention time as well as mass fragmentation characteristics. However, the sensitivity may be less than in the LBA. And by less, I mean you have a lower limit of quantitation. Now, often you can get around the sensitivity issues of direct digest and the specificity issues of ligand binding assays by combining them into a hybrid immunoaffinity LC-MS-MS assay. The difficulty here, or the trade-off that you have, is that now you have a much more complex assay that you have to develop and test all of the steps for. Here at Aliri, we are an expert CRO in direct digest. We have multiple assays online that have either been transferred into Aliri or that we have developed ourselves for direct digest LC-MS. We are currently bringing online and have analyzed samples for several hybrid immunoassays, LC-MS, and very soon we're going to be bringing LBA assays online as well. Now, when you talk about mass spec, both of these types that are on this slide talk about direct digestion, which leads to a big question in quantitative protein analysis. Are you going to do it top down or bottom up? Top down protein analysis is fairly difficult because you're trying to take the entire protein and make that fly down your mass spec. And that can be very difficult. As this cover from Angewandte Chemie in 2022 shows, it's like taking an elephant, strapping a bunch of helium balloons on it and making it fly. It's possible because you can usually put a large number of charge states on that mass in order to get it in within the range of your mass spec, but it is very difficult. A typical assay of top-down proteomics will start with your matrix, some kind of extraction of that protein, whether it's by gel or LC, and then injection on the mass spec where you can then fragment it, analyze the data, and get quantitation. The alternative, and the one that we at Aliri usually use, is bottom-up protein analysis. A typical experiment for bottom-up is taking the matrix, either extracting the protein or digesting the entire matrix. And digestion is taking that protein and breaking it up into the individual pieces or peptides or smaller pieces of the protein, and then separating it by LC, injecting on a mass spec in the determined range of the mass spec, and fragmenting it, analyzing the data, and then achieving your quantitation. The important difference between top-down proteomics and bottom-up proteomics is that top-down proteomics is a direct analysis of your protein. So you're looking at your analyte of interest directly, whereas in a bottom-up type quantitation, you are looking at a surrogate quantification because you are taking that protein and breaking it up into smaller surrogates that are signatures of that protein and quantifying it that way. So what does a typical bottom-up workflow look like? If you come to us with a FASTA sequence or just a protein of interest and say, hey, I want to quantitate this in my matrix, whether that's mouse or monkeys or humans, we'll start with an in silico analysis on the computer where we'll digest the protein and break it up into various peptides. We'll identify if those peptides are unique, determine some of the physiochemical properties of it before we even go into the lab. We'll then do a sort of a standard digestion, breaking up that protein, and do an IDA analysis, which I'll talk about in more detail later, in order to determine which peptides are going to be characteristic and have a good, strong signal. You can iterate this back and forth between looking at the computer and looking at your results on the mass spec, usually using a time of flight type instrument, before you then have to start building your actual assay. The assay, when you look at the initial data, is where you'll start to consider, do I need to do a pre-digestion cleanup? This would involve antibody culture, protein precipitation, something like that. If your protein is in high enough concentration, you can usually skip this step and move directly into the digestion, which is going to involve several steps: denaturization and reduction of the protein, alkylation of any free thiol or other reactive groups in that protein, then followed by digestion, usually using a trypsin reagent. After digestion, then you start thinking about your post-digestion cleanup. This can involve things outside the mass spec or the HPLC mass spec, such as solid phase extraction using anion exchange phases, or just simply injecting it onto the HPLC, getting appropriate separation, and then you move on to the detector. In the detector, you're going to be looking at the fragmentation of those peptides in order to identify it conclusively by looking at various Y and B ions. You can also use a summation of these fragments in order to help improve your signal. Now, a client coming to Aliri could enter the stage at any point in this process. They could come to us with a protein and say, hey, we're going to be looking at this in mouse, and we could start there. Or they could come to us with a completely developed method, and we could bring it online. With that, now, I'm going to dive into some of the details of each of these steps for the remainder of the webinar. The first part is the in silico analysis, or how to find what you're looking for. Overall, this is taking the FASTA sequence or the individual amino acids lined up in the protein and doing a tryptic digestion in the computer with it. This produces a series of candidate peptides. However, you have a long list of peptides for your protein of interest. You need to know if that peptide is unique to your protein or a signature for your protein, particularly in the matrix that you're going to be analyzing. And that's where a blast analysis, either using the NIH or UniProt, comes into play. You can conduct a blast analysis with that peptide and identify, hey, this is going to be unique. Once you have a large list, hopefully, of unique peptides, you move on to calculating those physical chemical properties and predicting the solubility, the pH responses, and the stability so that you have an idea before you get into the lab of which peptides are likely to work. The final step in the in silico analysis, and this is mostly just to help speed up the downstream effort, is to sort of look at what fragments your unique peptides will create so that then you could extract those from the time of flight data in order to quickly identify, hey, my peptide comes out at this portion. The identification of multiple fragments at a single retention time really helps support that that is the peptide that you're looking for and helps you decide which one to use. Now I'd like to take a small detour into a tool for characteristic or signature peptide identification. There are two methods that high-resolution time-of-flight instruments can use quite regularly. The first is swath data acquisition, and this is information-independent triggering. So it takes small time bins and looks at a whole bunch of the different peptides in it. And this is a great method if you're trying to identify the entire proteome of a system. However, when we're doing quantitation, we don't need the entire proteome. We want the most significant peptides for our characteristic protein. And that's where an IDA type workflow will work. And in this one, it only looks at fragmenting the top 10 or 15 most abundant signals. That helps you identify which peptides are the most significant and will produce the best signal in your system. Importantly, bottom-up quantitation is quantitation of a protein using the signature surrogate peptides. A signature peptide is unique to a protein of interest in the matrix that you're looking at, while a surrogate peptide is used to quantify the protein. The ideal situation, and the one we're always looking for, is a surrogate peptide that is a signature of the protein of interest. One other thing you're looking for when you're looking at the signature peptide is to avoid cysteine and methionine as amino acids in your peptide sequence. And that is because cysteine and methionine can be reactive during the digestion and handling of the sample, and you don't want modifications of your peptide once you have digested it. When you're looking at fragments of peptides, so your peptide comes off the LC column, goes into the instrument, and it hits the collision cell, they will often fragment into predictable Y and B ions. And those are simply where the charge is located on the peptide after fragmentation. So a Y ion of the fragment is on the C terminus, as highlighted in this figure here, for the Y2 being the DE of our peptide. So now we've done a whole bunch of work in the computer, and we want to get to the lab, and we want to digest things. So the important thing to remember is that everything we've done up to this point has been theoretical. However, as my co-presenter Shane likes to say, you can theorize all you want, but until you actually step into the lab and do the work, you don't know what the outcomes are going to be. So how do we actually get from a protein to a peptide, and how we break that up? The first thing you need to do is take that tertiary structure of the protein and break it up, unfold it, and denature it into the long primary structure. Part of that is going to be reducing any dithiol bonds in it. Disulfide bonds help maintain that tertiary structure, and by reducing them with a reducing agent such as DTT, you can produce free thiols. However, free thiols are incredibly reactive, could reform and help, and will make it more complex when you get to the digestion stage. The next step is going to be alkylate those free thiols with some kind of an alkylating reagent, and then you start the digester, breaking up that protein into the various small sections or peptides. The final one is to stop the reaction after the digestion and then perform any post-digestion cleanup and detection, either with the solid phase extraction, but definitely with an HPLC and a mass spec. So to reiterate, the first step is to denature the protein. And this is important because you're opening up the protein for trypsin to access the primary structure. While you're denaturing, you're going to reduce it or break the disulfide bonds to help maintain that usually help maintain that tertiary structure and can prevent it from opening. Once reduced, those free thiols need to be passivated because they are reactive and can refold the protein spontaneously over time. Once you get to the digest, you will usually use trypsin. Trypsin is incredibly specific, cleaving at arginine and lysine, and produce very consistent signature peptides from experiment to experiment. Now, the grade of trypsin you use is going to be important. If you have a protein that's in high abundance in your matrix, you can get away with a simple porcine trypsin from Sigma Aldrich. It's a natural trypsin collected from pigs, and it will do the work for you. However, if your protein is rarer, in lower concentration, or it's a protein that doesn't like to denature and it has some difficulty doing that, you may need higher concentration or higher quality trypsin, such as trypsin gold or trypsin platinum from Promega. If your protein is particularly finicky, you may even need a lyse mix, which will improve the peptide digestion over trypsin alone and increase recovery and better quantitation. Some notes of when you're doing a trypsin digest. Often in the denaturization step, you will add in additional components to help unfold that protein, such as glycine hydrochloride. However, if you use glycine hydrochloride, it is a strong inhibitor of trypsin, and so you will not be able to actually digest your trypsin. If your pH is too low, your trypsin will not function. You need a pH above 7.5. The final condition that people will often use in a digestion when they're denaturing is a strong urea concentration as a chaotropic reagent to break up that protein. However, if your urea, when you add the trypsin, is greater than 1 molar, you're going to have difficulty breaking up that protein. So now you have an assay, you have a digest. You need to get it over to the instrument to separate out the peptides and detect it, and that's going to be on an LC and mass spec. When we start with the LC, we're looking at the mobile phases first. Almost universally, 0.1% formic acid in water and 0.1% formic acid in acetonitrile are used. At the very beginning of this, when you're digesting your recombinant protein and just simply trying to find which peptides are present and where they are, you'll use this kind of an LC gradient, which is an exploratory one. It's very long, it's a very shallow gradient, and you're trying to separate out every single peptide that you can. Once you've identified your peptide, you'll move to this more typical gradient, which is, again, fairly shallow gradient, but it's significantly shorter before you clean off the column and re-equilibrate it for the next injection. LC-MS is naturally built for multiplexing, where you can monitor multiple peptides or multiple proteins and their signature peptides in a single assay. And in that case, you may be looking at multiple gradients in order to identify both of those peptides before you clean off and re-equilibrate. If your peptide has a lot of interferences around it, you may need to move to a more targeted gradient, where you start with an initially very steep gradient and then slowly elute to separate out that peptide from all of the other compounds that would be coming out before you clean off the column and re-equilibrate it. Of course, the LC gradients and the mobile phases are only part of an LC system. The other important part is the stationary phase. When you're looking at stationary phases, it's a good idea to use a peptide-optimized C18. These have very low carryover and less column interferences. Part of this is various manufacturers use very different substances to prevent any peptide-to-column interactions, such as Phenomenex here, which uses a biocompatible titanium, and Waters columns usually use a proprietary polymer to prevent those peptide column interactions. A 100 millimeter column is important, as your digest is not just your peptide of interest. It's composed of thousands of peptides, even if you're only seeing the one on the mass spec. So you want to have a significant column, a longer column, in order to properly separate out all of those ones before they get to the mass spec. That brings me to the mass spec, which is our detector here at Aliri. We are experts in using mass spec for small molecules as well as these peptides and macromolecules. As a reminder, your digested samples are very dirty because you've added a lot of reagents to the digest, especially if you haven't done any kind of post-digestion cleanup. So we need to do everything we can to keep that front end clean. This will result in better uptime of the instrument, better sensitivity for a lower limit of quantitation, as well as better data overall over the entire linear range. We can do this 3 primary ways. The first is using an integrated valve. This will allow you to select only what you need to see. So the salts coming off, usually at the very beginning of an injection, can be shunted to waste. You move your flow over to the mass spec while you need to look for your peptide. And then before you clean off the column, you shift it back to waste. And that helps prevent significant amounts of non-analyte materials hitting the detector. The second is using a scheduled ionization. So on a typical mass spec here, you have the front end, which pulls the ions into the instrument. Your Q1 selects the precursor mass. It is fragmented in Q2, and then you detect those fragments in Q3 or select for them before they hit the detector. In a scheduled ionization, you're going to shut down the front end of this instrument to prevent any ions coming into the rest of the instrument and therefore keeping it much cleaner. The final component that we can do to help keep your mass spec clean, to have better data overall, is maintain a high curtain gas while you are trying to detect. This is beneficial because it'll push neutral compounds that could foul the detector away from entering the orifice and going down the entire stream. With that, you have an assay. However, as my dad likes to say, no job is as easy as it ought to be. So there are going to be problems that crop up, and let's explore some of how we optimize this digestion and what to do when things start to go wrong. The first is digestion efficiency, and this is looking at how much protein becomes peptide. It's important because a low efficiency means you're going to end up with a very high lower limit of quantitation, especially if you're needing that sensitivity. And this is the main marker for assay optimization. There are a couple of things you need to know before you can perform this experiment, such as how many peptides are produced from the protein. For example, an IgG molecule may produce 2 signature peptides for every protein, and it's important to correct for that. Finally, you need to know the molecular weight of the protein and the peptide so that you can account for and make the concentrations match at the end of the experiment, and you can get an easy to interpret efficiency. Various corrections you need to make for are the purity of the protein and the peptide, as well as any dilution or volume changes through the assay. When you conduct this, you're going to have at least one well with your protein of interest in a known concentration, and you're going to have at least one well that's completely blank. You'll run both that blank and the protein of interest through your digestion until you get to the quench. And then at the quench, to the blank well, you're going to add an equivalent amount of peptide that should be produced if all of your protein is digested. From there, you can go to a post-digestion cleanup and LC-MS in order to quantitate how much of your protein became peptide. Of course, if your digestion efficiency is reasonably good, but you're still not hitting the lower limit of quantitation you want, then you may start turning to pre-digestion cleanups, such as immunocapture. It's a pre-digestion purification method and comes in 2 main forms. First is streptavidin binding to biotin, which is a common one used in the LBA field. And the second is using protein A, which will bind to IgG or other immunoglobulin heavy domain type molecules. This will help clean up your baseline and completely remove many of the interferences that might be impacting your assay. In order to perform one of these captures, you would bind your antibody to your analyte protein. Then you would take the streptavidin tag, the biotin tag, and then bind that to a streptavidin magnetic bead, for example. This could also be to a plate or to an on-tip streptavidin. You would then pull those beads over to a magnet, wash away a lot of the interferences, release it from the magnet or from the streptavidin bead, and then move on to the digestion. This is very helpful because you can then reduce the concentration of the reagents going into your digestion. And if you're needing to use very expensive digestion reagents, such as the trypsin lysate, it will help reduce the cost of the assay overall. It should make the baselines of your LC-MS much cleaner and help improve your signal at the bottom of your calibration curve. Here at Aliri, we have quite a few different ways of doing this. I mentioned magnetic beads in particular. This uses the basics of LBA on a magnetic bead, so you have an increased surface area compared to a well or a plate binding. We can also do automatic liquid handling, washing of those beads using a Hamilton Nimbus or Starlet. The final way that we have developed is using the Biotage streptavidin phi tip captures. So this is a tip that goes on an automatic liquid handler with a resin inside that has streptavidin on it. The general process is you can capture your sample, wash it to remove all those interferences, and then elute it before proceeding on with the digestion. And all of this can be done using an automated liquid handler so that an analyst is not tied up to run the analysis. Of course, pre-digestion cleanup may only get you so far, and you might want to look at a post-digestion cleanup. And this is going to be a solid phase extraction. The main principle of this is to remove the salts and other interferences that are not peptides. So if you have proteins that you're not interested in that didn't digest very well, it should remove those and help decrease the suppression or any interferences at your retention time for your signature peptide. A digestion is usually run at a pH of 8 to 8.5. As long as it's above 7.5, it should work. So you may be able to load it directly on an AX plate, depending on the nature of the charge pH response of that peptide. If it is not amenable to that, as in this example, you can quench it with formic acid to bring the pH down to 3 to 3.5, and then load on a strong cation exchange plate. There is one special type of cleanup, solid phase extraction that we have in-house here at Aliri, and that's using the EvoSep. This is an SPE on a tip using a C18 phase. It's very beneficial because it's sample cleanup and nanoflow chromatography all in one step. And the one other benefit of it is that it uses nanoflow or very low microflow, which can help improve your separation and sensitivity. And we've developed several assays for the EvoSep on our high resolution instruments. Now, the final thing I'd like to talk about before I hand over the presentation to Shane is quantification. There are multiple ways you can quantify your protein of interest. The first is using a surrogate peptide of a signature peptide, This is using the peptide as the calibrator and control samples. The one complication with this is that every time you do an entire run, you will need to include a digestion efficiency or positive control in that run so that you can correct for any lack of 100% conversion. An alternative to that is to do a direct digestion of the protein to the signature peptide. And this is still considered a surrogate quantification because, again, you're not looking at the protein as a whole, but as peptides produced from the protein for quantification. Now, the benefit of this is that your controls are inherently included in your digestion efficiency in all of the standards and quality control samples, but it might cost a bit more since you need a lot of recombinant protein to use as your standards and control samples. The final consideration in quantification is your internal standard, which will correct for injection-to-injection variability on the HPLC mass spec and any post-digestion cleanup you may be using. There are internal standard proteins that have heavily labeled amino acids incorporated into them, available from Sigma Aldrich for some of the common proteins. However, those tend not to be very useful, and we will tend to move toward an internal standard peptide that has a heavy label on the first or the last residue. So then you're looking at the Y and B ions of a heavy label and using that to correct for any changes. This plot here on the right is showing a metric plot or each injected sample for a real analysis that we did, showing some of the variability that the internal standard helps correct for. So in general, the development of a protein digest method requires multiple steps. That in silico digestion removes a lot of the lab work, hunting for likely signature peptides to use for your surrogate quantification. The digestion itself has multiple steps, each of which can have significant changes to the outcome of the assay. If your headspace is that you treat a digested sample as just a very dirty small molecule, you're headed in the right direction because you can't usually precipitate out a peptide. While solid phase extractions may help clean it up. Pre-digestion concentration and cleanup can help reduce many of your downstream issues, as well as post-digestion cleanup can help improve your signal and help remove significant noise. So with this background on the digestion and quantification of peptides, I'd like to introduce Shane to talk about some of the regulatory considerations when you're bringing these protein methods to a regulated study.
Great. Hey, Matt. Thank you for that introduction to protein LCMS development. To follow up on protein LCMS method development, I'm going to discuss regulatory method validation and sample analysis for these styles of assay. Go to the next slide, please. When it comes to protein analysis by LCMS, the experiments and acceptance criteria used for method validation will differ depending on the workflows used, as Matt has described. For instance, if using immunocapture, the criteria will be more aligned with LBA acceptance, whereas if you are directly digesting the protein without capture, the criteria may be closer to a small molecule assay. Next slide, please. For the preparation of calibrators and QCs for protein analysis, there are several options. If you are preparing your calibrators with a surrogate peptide, but preparing your QCs with a recombinant protein, one benefit is that the QCs will mimic incurred samples, but this approach may require a correction factor since digestion efficiency will not be accounted for. Alternatively, when recombinant proteins are used to produce both the calibrators and the QCs, the assay will best mimic the treatment of incurred samples, but an issue may arise if the digestion efficiency is variable from run to run, and this issue will likely result in the loss of signal at the LLOQ of the assays for runs where the digestion efficiency was low. Finally, when surrogate peptides are used for preparing both the calibrator and the QCs, the benefits are that they will represent 100% digestion efficiency, but the disadvantage is that the true digestion efficiency of the incurred samples may be difficult to determine. Next slide, please. Okay, so a lot in this table. So this table lists the various experiments required for different assay designs based on the criteria listed in the M10 guidance. For a protein LCMS assay, the experiments and acceptance criteria will be a hybrid and include experiments and acceptance criteria from both LBA and small molecule type studies. Prior to validating a hybrid mass spec method, acceptance criteria and experiments should be determined a priori in a method validation plan based on the robustness of the assay determined in method development. In the case where surrogate peptides are used for calibrators and QCs, the acceptance criteria and experiments will closely follow a small molecule assay. In contrast, when immunocapture is used and calibrators and QCs are prepared with recombinant protein, the criteria will likely follow an LBA acceptance criteria. Next slide, please. For example, this slide shows the typical method validation acceptance criteria and experiments for an immunocapture LCMS assay. For this validation, selectivity, dilution integrity, and matrix effect experiments would be performed following mass spec experimental design, whereas the accuracy and precision criteria would follow LBA acceptance criteria. As with all method validation, the experiment and acceptance criteria should be pre-established and documented in an SOP or method validation plan before starting your validation experiments. Next slide, please. Sample analysis using immunocapture LCMS would use the same acceptance criteria used for small molecule analysis, except the accuracy of the assay would likely be expanded to match LBA criteria. For these studies, ISR would be performed, but again, the LBA acceptance criteria would be used, which is plus or minus 30% difference. For sample analysis, samples would be run in singlet and internal standard response would be monitored to determine if there is systemic variability of the IS response in incurred samples. Next slide, please. When proteins are measured in tissue, the level of validation will depend on the sponsor's need. Tissue assays can be validated as close to GLP as required based on the intended use, but all stability experiments will be performed in tissue homogenate. This slide lists the typical experiments included in a near GLP tissue validation. Alternatively, tissue homogeneity analyzed after dilution with plasma, typically in a 1 to 10 ratio using a fully validated plasma method, is a common approach to measure protein level to support bioanalysis studies. Again, the level of validation and the validation design will depend on the sponsor's need. Next slide, please. For protein biomarker validation, the level of validation will be determined by the context of use. For example, a predictive biomarker in early discovery would require a minimal method qualification, whereas a label claim biomarker would require a full validation. If the biomarker information will be used to adjust the dose of a medication being given to a patient, the level of validation would follow CLIA or CAP criteria. Regardless of the amount of validation deemed necessary for a biomarker assay, it is important to have the experiments and their criteria pre-established before conducting any studies. Next slide, please. And with that, I'm coming to the end of the webinar. So I wanted to say thank you to all the participants who logged on to watch this today. I also wanted to say thank you to our staff at our various sites, our sites in Utah, Colorado, and France, and their help for preparing this webinar. And at this point, me and Matt will take any questions that you all may have. Thank you very much.
Fantastic. Thank you all for that presentation. It looks like a few of the audience members have already submitted questions, so we're going to jump right into the Q&A session. So the first question we have is, what kind of turnaround does Aliri have if we start with just the protein of interest in matrix?
So it'll depend quite a bit on the characteristics of the protein. In general, we say probably 4 to 6 weeks for complete turnaround from starting with a protein of interest through complete assay development and saying, here we go, we can take samples now. I think, Shane, you would agree with that kind of a timeline?
Yeah, definitely. That is within the scale of what we would do. Again, always the complexity and the LLOQ is always the very much a driving factor.
Yes. So like if it's a very high concentration protein in your matrix, usually you can turn that around much faster because the LLOQ is less demanding. Whereas if you're having a very unique protein at very low concentrations, then you're going to have to start developing some of those alternative workflows like the pre-digestion cleanup or the post-digestion cleanup.
Yeah, agreed. Yeah.
Thank you. It looks like the next question is, how long does a trypsin digestion need to be?
So this again depends on the nature of the protein and how well it denatures in that particular step. And the denaturization step will become very important prior to the trypsin digestion. If your protein does not digest well, you may need very long times. And that's where you're going to start looking at overnight, so 16 hours to 20-hour digestion periods. That's also where you start needing higher quality trypsins because trypsin can digest itself over that period. So if you can get away with a 2-hour digestion, you can usually also get away with less expensive trypsin. Whereas if you start going into those longer overnight digestions, that's when you start considering higher quality, the trypsin gold, the trypsin platinum, or even the lyc trypsin mixes.
Yeah, and I might just add on there, length of time, it's always important to optimize that to kind of fit into what is a normal laboratory workday as well.
Yes.
It's never great to have people coming in at midnight to quench a reaction. So, there is always kind of a bit of a balance there to kind of have a decent work-life balance as well. But obviously, you got to do what the science dictates and do the right amount of digestion.
Absolutely. And to stay with that kind of theme of trypsin, this next question asks, how does Aliri decide what grade of trypsin to use?
So I sort of talked about it just now, but it depends quite a bit on how long your digestion is going to be and the complexity or the difficulty of working with your protein of interest. A lot of that can be determined early in the development process when you're looking at that in silico analysis.
Yeah, and I might add there are things like RAPIGES surfactants that can be added to somewhat help with digestion. So maybe you can get away with a lesser grade of trypsin. That's
Yes.
some techniques that are out there as well.
Thank you. We have another question that asks, how does picking a fragment mass at a higher mass to charge ratio than precursor mass help the sensitivity of the analysis?
Okay, so this is starting to get into the nitty gritty of the mass spec development, which is great. I love it. When you have a peptide, they usually come off of the LC column with a +1 or a +2 charge, sometimes even +3. When you have that high charge, your mass to charge ratio is going to be greater, or rather the number coming onto the mass spec is low. Low mass to charge ratios tend to be where a lot of the noise is. So if you can start with a +2 charged peptide, let's say 400 m/z, and then fragment it and increase that or decrease the charge to increase the mass to charge ratio to a higher level, you can move that fragment out of a lot of the noise that would come at the lower m/z values. And if you can move it out of the noise, then you start getting cleaner baselines and you can achieve lower levels of quantitation.
Thank you. And the next we have is for validating a biomarker assay. What guidance would you follow?
Yeah, it's a great question. So prior to the release of the new M10, I guess it's not new, it's been out over a year now, but release of the new M10 bioanalytical guidance, the 2018 FDA guidance covered biomarkers, but they do not appear in the M10 guidance. So at this point, I think the best, use is to go back to the 2018 FDA guidance that covered, bioanalytical validations and included a section on biomarkers. So you would use the information from there. But I think the most important thing is really understanding what it is you're trying to accomplish with your biomarker, and make sure that the tests you design for your validation really show that the decision making for that biomarker is going to be good with the assay, so that you can actually make decisions. So regardless of the guidance you're following, you really need to do good science and make sure that the data you're reporting is reliable and it can be used for decision making process, and that regulatory reviewers are going to agree with your decision making. So
Absolutely. And it looks like you guys are pretty popular because we keep having questions coming in. So the next one is, as you said, you only choose a few peptides to quantify. How do you prioritize which peptides from a protein to quantify?
So this is going to come from that initial workup of the in silico analysis that you do. When you do the in silico trypsin digest, you're going to have a huge number of candidate peptides. From there, you'll run the blast analysis to ensure that those peptides are not present in other proteins or pieces in the matrix. So that's the main way you're going to select which ones to use. After that, you're going to look at actual data on the mass spec. Which ones are giving you the sensitivity and the selectivity that you need? Because you don't want to choose a peptide that you can't see, essentially. So it's a twofold component. Looking at that blast analysis to make sure that your peptide is a signature for the protein you're looking at, and then also seeing which peptides actually give you some data and sensitivity that you need.
Awesome. And the next question we have is, how much faster is using the EvoSep versus more traditional SPE cleanup workflow?
So this is sort of a two-part question. It's a little bit more complicated than I think the asker meant. The EvoSep, from an analyst in the lab's perspective, is much faster than a traditional SPE, because you can just load the tips, which have the C18 phase on them, very quickly. You can wash them very quickly. It takes about an hour to do a full plate, whereas a traditional SPE may take longer than that, depending on how many washes or what kind of conditions you need in order to work with that plate. The benefit that the EvoSep provides is when you get to the mass spec. Because it uses nanoflow, the time on the instrument is actually longer usually than your traditional SPE, which goes on a standard LC. The EvoSep uses nanoflow, which can provide a much lower level of detection, as well as better separation between peptides of interest. So you get a much cleaner signal than you might get with just a standard SPD.
And it looks like our last question for today is, if you take the surrogate peptide approach and the titer criteria is applied, what would be used for ISR acceptance criteria, since it would be done with the intact protein?
Yeah, that is a great question. So, for ISR acceptance, in a situation where you're using, surrogate peptide calibrators and QCs, but your samples are truly, protein, expanding that acceptance criteria to be more in line with LBA, I think would be easy to justify, because really when you're looking at the incurred sample analysis, you're looking at how does the sample perform, how does it digest. You're not really looking at calibrator and QC accuracy and precision at that point. So expanding that criteria out to match LBA, I think would be justifiable. Again, I think with any of this, it always depends on, how good is your method, when you were doing development. Do you feel like you have a very, precise, rugged, robust method? Then you might be able to get by with tighter criteria. But if you're running a more complex matrix, if you're doing this in tissue where you have a lot more proteins and around, you might find that you need to expand that criteria. So this is really, I think the important thing here is with any study, it's just to make sure that what you're going to do, ahead of time, you put it in the sample analysis plan and you follow that criteria and you live with the results based around that. So great question. Thank you very much.
Great. Well, it looks like those are all the questions that we have today. So thank you for your responses. I'd like to really thank our speakers today, Shane, Ed, and Matt, for that really insightful presentation, and our sponsor, Aliri Bioanalysis, for making this event possible. On behalf of Aliri Bioanalysis and Sightline, I hope everyone has a great remainder of their day, and thank you for watching.
Thanks, everyone.
Yeah.